Good afternoon everybody. I J. Sureka, Head Department of Biochemistry. Today we are going to see about restriction fragment length polymorphism. Principle of Rflp. Rflp is an enzymatic procedure for separation and identification of desired fragment of DNA. Using restriction endonuclease enzyme, fragments of DNA is obtained and the desired fragment is detected by using restriction probes. Sudden hybridization using restriction endonuclease enzyme for isolation of desired length of DNA fragments is an example of Rflp. This is the overall step of Rflp. In step 1, restriction digestion will take place. Extraction of desired fragment of DNA using restriction endonuclease. The enzyme restriction endonuclease has specific restriction site on the DNA, so it cut DNA into fragments. Different size of fragments are generated along with the specific desired fragments. Steps 2 is an gel electrophoresis. The digested fragments are run in polyacrylamide gel electrophoresis or agarose gel electrophoresis to separate the fragments on the basis of length or size or molecular weight. Different size of fragments from different bands. The step 3 is an denaturation step. The gel is placed in sodium hydroxide solution for denaturation so that single-stranded DNA are formed. Step 4 is an blotting. The single-stranded DNA obtained or transferred into charged membrane, that is nitrocellulose paper, by the process called capillary blotting or electroblotting. Step 5 is an baking and blocking. The nitrocellulose paper transferred with DNA is fixed by autoclaving. Then the membrane is blocked by using bovine serum albumin or casein to prevent binding of labeled probe non-specifically to the charged membrane. Step 6 is an hybridization and visualization. The labeled Rflp probe is hybridized with the DNA on the nitrocellulose paper. The Rflp probes are complementary as well as labeled with radioactive isotopes, so they form color band under visualization by autoradiography. Applications of Rflp. Genome mapping helps in analysis of unique pattern in genome for organism identification and differentiation. It also helps in determining recombinant rate in the loci between restriction site. Genetic disease analysis. After identification of gene for particular genetic or hereditary disease, that gene can be analyzed among other family members to detect mutated gene. DNA fingerprinting, that is forensic test, it is the basis of DNA fingerprinting for paternity test, criminal identification, etc. Thank you.